Introduction. Welcome students to today's lecture on cell biology and the intricate mechanisms governing cell cycle regulation. Today, we will delve into the fascinating world of checkpoints and their vital role in maintaining the integrity and precision of the cell cycle. Starting with the regulation of the G1 checkpoint and concluding with the complex checkpoint adaptation mechanisms, we will explore the detailed intricacies of each topic. So, let's begin our journey into the world of cell cycle checkpoints. The cell cycle is controlled by checkpoints to prevent abnormal cell proliferation. The G1 checkpoint monitors cell growth and nutrient availability before committing to DNA replication. Key proteins, such as cyclins, CDKs, and ARBA, regulate the G1 checkpoint, preventing DNA replication until the cell is ready. In the S phase of the cell cycle, DNA is replicated to prepare for cell division. However, errors can occur, so cells have a surveillance mechanism called the S phase checkpoint. This checkpoint detects DNA damage or incomplete replication and stops DNA synthesis for repair. Proteins like ATR and ATM are key players in this checkpoint, triggering DNA repair pathways. In the cell cycle, the G2/M checkpoint ensures accurate progression into mitosis. During this checkpoint, the Cyclin-B/CDK1 complex is activated and regulated, playing a crucial role in orchestrating mitotic events. Regulatory proteins like WE1, MIT1, and CDC25 control the activity of this complex to prevent premature entry into mitosis. During mitosis, when the duplicated chromosomes segregate into two daughter cells, it is crucial to ensure the proper attachment and alignment of chromosomes to the spindle apparatus. The spindle assembly checkpoint monitors this process, delaying anaphase until all chromosomes are correctly attached to the spindle microtubules. If attachments are incorrect or incomplete, the checkpoint proteins, such as MAD1, MAD2, and BUBBER1, generate a signal that prevents the activation of the anaphase-promoting complex/cyclosome, APC/C, an essential regulator of mitotic progression. This delay allows sufficient time for error correction and ensures equal chromosome distribution to daughter cells. DNA damage poses a significant threat to genome stability and cell viability. To counteract this challenge, cells employ various mechanisms to sense and repair DNA lesions before progressing through the cell cycle. This includes the activation of checkpoint kinases, such as ATM and ATR, which phosphorylate downstream targets involved in cell cycle arrest, DNA repair initiation, and program cell death if damage is beyond repair. Additionally, DNA repair pathways, such as base excision repair, nucleotide excision repair, and homologous recombination, are activated to restore DNA integrity. These sophisticated mechanisms provide an opportunity to repair DNA damage and maintain genomic stability. The p53 tumor suppressor gene is crucial for maintaining genome stability. It regulates genes that control cell cycle arrest, DNA repair, and apoptosis in response to DNA damage. Loss of p53 function is common in cancer cells, leading to uncontrolled growth and resistance to cell cycle checkpoints. The ubiquitin proteasome system, UPS, is a critical regulatory pathway involved in cell cycle control. It selectively tags proteins with a small protein called ubiquitin, targeting them for degradation by the proteasome. In the context of the cell cycle, the UPS plays a pivotal role in regulating the timely degradation of key regulatory proteins. For instance, cyclins and cyclin-dependent kinase inhibitors, CKIs, are continuously synthesized and degraded throughout the cell cycle, ensuring their precise temporal control. Aberrant cell cycle regulation lies at the heart of cancer development. Dysregulation can manifest in various ways, such as mutations or altered expression levels of key regulatory proteins, disrupted signaling pathways, or malfunctioning DNA repair mechanisms. These alterations can lead to uncontrolled cell proliferation, evasion of cell cycle checkpoints, and genomic instability, all hallmarks of cancer. Understanding the molecular basis of cell cycle dysregulation in cancer is crucial for developing targeted therapies aiming to restore normal cell cycle control. The activity of cyclin-dependent kinases, CDKs, is precisely regulated through a series of phosphorylation and dephosphorylation events. Cyclin binding triggers conformational changes in CDKs, promoting their activation. Additional phosphorylation events by CDK activating kinase, CAK, increase their kinase activity, enabling phosphorylation of downstream targets involved in cell cycle progression. Conversely, CDK inhibitors, such as P21 and P27, block CDK activity by binding to the cyclin-CDK complex, preventing substrate phosphorylation. The delicate balance of phosphorylation and dephosphorylation events ensures proper CDK activity throughout the cell cycle. Beyond phosphorylation, post-translational modifications, PTMs, play essential roles in cell cycle regulation. These modifications include ubiquitination, acetylation, methylation, sulmylation, and glycosylation, among others. PTMs can modulate protein stability, localization, and activity, providing an additional layer of complexity to cell cycle control. For example, the degradation of cyclins during mitosis is regulated by ubiquitination, while acetylation of histones contributes to the regulation of gene expression during the cell cycle. Epigenetic alterations, such as DNA methylation, histone modification, and chromatin remodeling, have emerged as critical players in cell cycle regulation. These modifications can influence the expression of genes involved in cell cycle progression, DNA repair, and checkpoint activation. Aberrant epigenetic changes can disrupt normal cell cycle control, leading to uncontrolled proliferation or inadequate response to DNA damage. The study of epigenetic modifications and their impact on cell cycle regulation provides valuable insights into the development of novel therapeutic strategies that target these alterations in cancer and other diseases. Checkpoint adaptation mechanisms allow cells to override cell cycle arrest induced by checkpoints in certain circumstances. Cells may adapt to prolonged arrest, allowing cell division to proceed despite incomplete DNA repair or other challenges. This adaptation mechanism involves the downregulation or inactivation of certain checkpoint components, enabling cell cycle progression. However, the precise mechanisms and consequences of checkpoint adaptation are still under investigation, and their understanding holds significance in cancer biology and therapy. Understanding the intricacies of cell cycle checkpoints is vital for medical students. Dysregulation of these checkpoints can lead to diseases like cancer. By studying the detailed mechanisms of checkpoint control, we can identify potential therapeutic targets for combating these diseases.